In the world of extreme survival, few creatures can match the remarkable resilience of tardigrades, also known as water bears. But in 2020, these microscopic extremophiles revealed yet another extraordinary capability that had remained hidden for centuries. They glow blue under ultraviolet light. Join us as we explore how a chance discovery at the Indian Institute of Science unveiled not just a biological curiosity, but a sophisticated survival mechanism that could inspire revolutionary applications in medicine, agriculture, and space exploration. Our story begins in 2020 at the Indian Institute of Science in Bangalore, India. Biophysicist Sandeep Eswarappa and his team were conducting what they thought would be routine experiments on tardigrades. They were studying a specific species called Paramacrobiotus, specifically the PBLR strain, which they had collected from moss samples growing on a concrete wall right in Bangalore. The researchers were investigating how these tiny creatures resist germicidal UV radiation. Already an impressive feat, when something unexpected happened. As they exposed the tardigrades to ultraviolet light, the microscopic organisms began to emit a distinct blue glow. This wasn't the result of any staining techniques or artificial manipulation. The tardigrades were naturally fluorescent, emitting blue light at wavelengths between 420 and 474 nanometers when exposed to UV light in the 335 to 350 nanometer range. What makes this discovery so remarkable is its completely serendipitous nature. Had the team not decided to examine their specimens under UV light, a method not typically employed in tardigrade research, this phenomenon might have remained hidden for many more years. Despite tardigrades having been studied by scientists since they were first described in 1773. To appreciate why this fluorescence discovery is so significant, we need to understand just how extraordinary tardigrades already were before this revelation. These microscopic animals, barely visible to the naked eye at less than a millimeter long, are perhaps the most resilient creatures on our planet. They've been found surviving in virtually every environment imaginable, from the deepest ocean trenches to the highest mountain peaks. Their survival capabilities border on the supernatural. Tardigrades can enter a dehydrated state called anhydrobiosis, where they reduce their water content to less than 3% and essentially suspend all metabolic processes. In this state, they can remain viable not just for days or months, but for decades. They've been subjected to the vacuum of space and survived. They've endured temperatures ranging from near absolute zero, minus 273 degrees Celsius, to over 150 degrees Celsius. They've withstood pressures 6 times greater than that found in the deepest ocean trenches. Perhaps most impressively, they can survive radiation doses thousands of times higher than what would be lethal to a human being. And now, with the discovery of biofluorescence, scientists have uncovered yet another remarkable adaptation in their already impressive survival toolkit. Eswarappa's team quickly realized that this fluorescence wasn't merely a biological curiosity. It served a critical survival function. Through careful experimentation, they discovered that the fluorescent pigments in the tardigrade's cuticle, or outer layer, act as a sophisticated protective mechanism. These compounds essentially convert harmful UV radiation into safer blue light, a process called fluorescent photoconversion. To test this protective function, the researchers extracted these fluorescent compounds and applied them to UV-sensitive organisms, including Sandeep elegans worms and non-fluorescent tardigrade species. The results were striking. Organisms treated with these compounds showed significantly increased survival rates when exposed to UV radiation. Quantitative analysis revealed that tardigrades with this fluorescent capability survived UV radiation doses up to 1 kilojoule per square meter with minimal damage, while non-fluorescent species experienced significant mortality at the same exposure levels. This translates to a survival advantage of approximately 60% in high UV environments, a substantial evolutionary edge. Unlike conventional sunscreen that blocks or reflects UV radiation, the tardigrade's approach is fundamentally different. Their fluorescent compounds don't just stop the radiation, they transform its energy into a different, less damaging form of light. This elegant solution allows them to neutralize the threat while potentially putting some of that energy to use. The protective compounds responsible for this fluorescence have been identified as a unique class of tetrapyrroles. These molecules feature a central ring structure similar to that found in chlorophyll and hemoglobin, but with specific modifications that enable their wavelength conversion properties. What's particularly impressive about these compounds is their remarkable stability. Spectroscopic analysis has shown that they maintain their functionality even after repeated radiation exposure cycles. Most synthetic fluorescent materials degrade under prolonged UV exposure, a phenomenon called photobleaching. But the tardigrades' natural compounds remain stable, suggesting an extraordinary molecular resilience. The distribution of these compounds throughout the tardigrade's body is equally fascinating. They appear to be concentrated in specialized organelles throughout the cuticle, positioned to maximize their protective effect while minimizing the metabolic cost of production. This represents a stunning example of evolutionary optimization at the microscopic level. The molecular arrangement allows for maximum UV absorption and efficient energy conversion, creating what amounts to a living, self-repairing sunscreen that never needs to be reapplied. The discovery of biofluorescence in tardigrades adds them to a growing list of organisms that utilize this phenomenon, including certain fish, amphibians, birds, and marine invertebrates. However, what makes the tardigrade case particularly fascinating is that it appears to be an example of convergent evolution, the independent development of similar traits in unrelated lineages facing similar environmental pressures. Genetic analysis of fluorescent tardigrades has revealed that their fluorescence mechanism evolved independently from that of other fluorescent organisms. The genes responsible for producing these tetrapyrrole compounds show no close homology with fluorescence-related genes in other species. This suggests that tardigrades developed this adaptation separately over evolutionary time, rather than inheriting it from a common ancestor shared with other fluorescent creatures. This evolutionary independence is especially intriguing because tardigrades have existed for over 500 million years, surviving all 5 mass extinction events that have occurred on Earth. The fluorescence adaptation may have emerged during periods of heightened UV radiation, such as during the Permian-Triassic extinction event approximately 252 million years ago, when ozone depletion led to significantly increased UV radiation reaching Earth's surface. Phylogenetic studies comparing various tardigrade species suggest that this fluorescence capability evolved approximately 150 to 200 million years ago, during the Jurassic period. This timing coincides with significant global climate shifts and habitat transitions, potentially driving selective pressure for enhanced UV protection. The adaptation appears most prevalent in tardigrade species that colonized semi-terrestrial environments like moss and lichen, where exposure to direct sunlight presented new challenges compared to their aquatic ancestors. What's particularly interesting is that the fluorescence trait shows a mosaic distribution across the tardigrade phylogenetic tree. This suggests it may have been gained and lost multiple times throughout their evolutionary history as environmental pressures shifted. Some lineages developed the adaptation when moving into high UV environments, while others may have lost it when moving to low-light habitats where the metabolic cost of maintaining the fluorescent compounds outweighed their protective benefits. This pattern of repeated evolution and loss speaks to the remarkable genetic plasticity of tardigrades and their ability to adapt to changing conditions over geological timescales. Recent research has revealed that the fluorescence capability of tardigrades serves additional ecological functions beyond simple UV protection. Field studies conducted in alpine environments have demonstrated that fluorescent tardigrade species show distinctive behavioral responses to varying UV intensities. When exposed to increasing UV radiation levels, these tardigrades exhibit negative phototaxis, actively moving away from the light source at rates proportional to the UV intensity. This behavior suggests the fluorescence may function as part of a sensory system, allowing tardigrades to detect and respond to potentially harmful radiation levels before cellular damage occurs. Laboratory experiments using specialized microfluidic chambers have shown that fluorescent tardigrades can detect and respond to UV radiation levels as low as 0.5 milliwatts per square centimeter, well below the threshold for DNA damage. This sensitivity enables them to seek shelter before exposure reaches harmful levels, providing an early warning system that complements their physical protection. What's particularly fascinating is how this adaptation varies between populations from different habitats. The fluorescence emission spectrum differs slightly between specimens collected from different elevations, with high-altitude tardigrades showing emission peaks shifted approximately 12 nanometers toward shorter wavelengths compared to lowland populations. This spectral tuning may represent fine adaptation to the specific UV profiles encountered in different environments, with high-altitude populations adapted to the more intense, shorter wavelength UV radiation typical at elevation. The discovery of these natural UV protective compounds has sparked significant interest in the biomedical field. Researchers at the University of California, San Diego have begun investigating tardigrade fluorescent compounds as potential ingredients for advanced sunscreens that could offer superior protection compared to current formulations. Unlike conventional sunscreens that need to be reapplied frequently and can cause skin irritation in some people, A tardigrade-inspired formulation might provide longer-lasting protection with fewer side effects. Perhaps even more promising is the potential application for treating photosensitivity disorders like xeroderma pigmentosum, a rare genetic condition that affects approximately 1 in a million people worldwide. Individuals with this condition are extremely sensitive to UV radiation and have a 1,000-fold increased risk of developing skin cancer. Current treatments are limited, but tardigrade fluorescent compounds could potentially provide a new therapeutic approach by converting harmful UV radiation before it can damage sensitive skin cells. The molecular stability of these compounds makes them particularly attractive for medical applications. Unlike many biological compounds that degrade quickly outside their natural environment, tardigrade fluorophores maintain their protective properties even when extracted and applied to other organisms. This stability suggests they could be incorporated into long-lasting medical treatments that require minimal reapplication. In materials science, the stable fluorescent properties of tardigrade compounds present opportunities for developing new UV-resistant coatings and materials. Engineers at MIT's Materials Research Laboratory have initiated projects to synthesize analogs of these compounds for application in spacecraft shielding, where protection from cosmic radiation remains a significant challenge for long-duration missions. Current radiation shielding for spacecraft is bulky, heavy, and often relies on simple mass to block radiation, an approach that comes with significant weight penalties for launches. A coating based on tardigrade fluorophores could potentially provide comparable protection at a fraction of the weight by converting harmful radiation rather than merely blocking it. This would be particularly valuable for long-duration missions to Mars or beyond, where minimizing spacecraft weight while maximizing radiation protection is a critical design challenge. The applications extend beyond aerospace. Electronic components, especially those in satellites and high-altitude drones, are vulnerable to UV degradation. Protective coatings inspired by tardigrade fluorescence could extend the operational lifespan of these components in high-radiation environments. Similarly, architectural materials exposed to sunlight, such as paints and plastics, could benefit from enhanced UV resistance, potentially extending their useful life by years or even decades. The unique wavelength conversion properties of these compounds could even be harnessed for energy applications, potentially converting otherwise wasted UV radiation into visible light that could be captured by conventional solar cells. Perhaps most intriguingly, the specific wavelength conversion properties of tardigrade fluorophores make them candidates for enhancing photosynthesis in low-light environments. Agricultural researchers have begun preliminary studies on using modified versions of these fluorophores to coat greenhouse materials, potentially converting unusable UV radiation into wavelengths optimal for plant growth. Plants primarily utilize light in the red and blue portions of the spectrum for photosynthesis, while much of the UV spectrum goes unused or can even damage plant tissues. By converting this UV radiation into blue light in the 420 to 474 nanometer range, precisely the wavelengths emitted by fluorescent tardigrades, these compounds could effectively expand the usable light spectrum for plants. Initial studies suggest this technique could increase crop yields by an estimated 15 to 20% in controlled environments without requiring additional energy input. This approach could be particularly valuable for vertical farming operations in urban environments, where maximizing plant growth per unit of energy is essential for economic viability. It could also benefit agriculture in regions with high UV but lower visible light levels, such as high-altitude farming communities in the Andes or Himalayas. The technology could even have applications for future space-based agriculture, where optimizing every photon of available light will be crucial for growing food on long-duration missions or extraterrestrial settlements. The discovery of biofluorescence in tardigrades represents more than just another fascinating adaptation in these already remarkable creatures. It exemplifies how much remains unknown even about organisms that have been studied for centuries. First described by Johann August Ephraim Gose in 1773, tardigrades have been the subject of scientific inquiry for nearly 250 years. Yet this fundamental aspect of their biology remained hidden until 2020. This revelation highlights the importance of serendipity in scientific discovery and the value of approaching familiar subjects with new techniques and perspectives. Had Eswarappa's team not decided to examine their specimens under UV light, a method not typically employed in tardigrade research, this phenomenon might have remained undiscovered for many more years. The story of tardigrade fluorescence reminds us that some of the most significant scientific discoveries happen not through careful planning, but through chance observations by researchers who are attentive enough to recognize the importance of unexpected results. It also demonstrates the value of basic research. studies conducted simply to expand our understanding of the natural world, without immediate practical applications in mind. While the initial discovery of tardigrade fluorescence wasn't aimed at developing new sunscreens or agricultural technologies, it has now opened doors to these and many other potential applications. As climate change alters our environment in numerous ways, understanding natural protection mechanisms against UV radiation becomes increasingly valuable. Ozone depletion and shifting weather patterns have already increased UV exposure in many regions, with potential consequences for both human health and ecosystems. The tardigrade's elegant solution to UV radiation, converting it rather than merely blocking it, offers a conceptual blueprint for developing more efficient and effective protection technologies. This approach could be particularly relevant as we face growing challenges related to UV exposure. Skin cancer rates have been rising globally, with UV radiation being the primary environmental risk factor. Agricultural crops face increased stress from higher UV levels, potentially threatening food security in vulnerable regions. Even marine ecosystems are affected, with UV penetration damaging coral reefs and disrupting the development of fish eggs and larvae near the ocean surface. By studying how tardigrades have solved the UV protection problem through fluorescent photoconversion, we may develop more effective approaches to addressing these challenges across multiple domains. Their solution, developed over millions of years of evolution, may prove more elegant and effective than anything human engineers could design from first principles. As is often the case, nature may have already solved problems we are only beginning to address. The story of fluorescent tardigrades reminds us that some of the most profound solutions to challenging problems may already exist in nature, hidden in plain sight within organisms we've long overlooked or underestimated. Despite centuries of scientific exploration, our planet still harbors countless undiscovered biological adaptations and mechanisms that could inspire new technologies or medical treatments. Microscopic organisms like tardigrades represent a particularly rich frontier for such discoveries. Their small size and the specialized equipment needed to study them in detail have limited our understanding of their biology, despite their ubiquity in environments worldwide. As new imaging technologies and molecular analysis techniques become available, we're increasingly able to peer into the microscopic world and uncover its secrets. The tardigrade's glow serves as a powerful reminder that even in organisms we thought we understood well, fundamental discoveries await those willing to look with fresh eyes and new methods. It also highlights the importance of preserving biodiversity, even among microscopic life forms that rarely capture public attention. Each species represents the outcome of millions of years of evolutionary problem-solving, potentially harboring adaptations that could address human challenges in ways we haven't yet imagined. As we continue to unravel the molecular mysteries of these microscopic marvels, we may find that the water bear's glow illuminates paths toward technological and medical advances that once seemed beyond our reach. From a chance observation in a Bangalore laboratory to potential applications spanning medicine, agriculture, and space exploration, the discovery of fluorescent tardigrades exemplifies the wonder and practical value of basic scientific research. These microscopic water bears, already famous for surviving the most extreme conditions on Earth, have revealed yet another extraordinary adaptation: the ability to transform deadly UV radiation into harmless blue light through specialized molecular compounds in their bodies. This protective mechanism, developed over millions of years of evolution, now offers blueprints for technologies ranging from advanced sunscreens and treatments for photosensitivity disorders to radiation-resistant materials for spacecraft and yield-enhancing coatings for agricultural greenhouses. As climate change increases UV radiation exposure worldwide, the tardigrade's elegant solution becomes increasingly relevant to human challenges. Perhaps most importantly, this discovery reminds us that despite centuries of scientific inquiry, nature still holds countless secrets waiting to be uncovered. The humble water bear, glowing blue under ultraviolet light, illuminates not just the remarkable ingenuity of evolutionary adaptation, but also the endless possibilities that await when we approach the familiar world with fresh perspective and open minds. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.